that Andre asked me to kind of share with you and try to explain in simple terms. So I'll try to do that. So paper is on a website called bioRxiv. And so bioRxiv is a place where scientists, before they sort of officially publish their paper and it is peer-reviewed, they put it out. So it's important to note. I don't know if you can see my mouse, but it's important to note this little a sentence here in the middle of the page that in light gray that says this article is a preprint and has not been certified by peer review. So that means basically that these are the findings as we have found it, but experts in the field have not had a chance to give their opinion on it and us to respond to their feedback and criticism to improve the work. So this is you have to basically say take everything that I say in the next half an hour as preliminary data. And so what this paper tries to present is, first of all, a new animal model with symptoms that are a little bit akin to the symptoms that we see in OGT-CDG patients. So this is only the second animal model of this disease, and we try a somewhat unusual approach to see whether we can rescue some of these OGT-CDG symptoms in this mouse by making use of another mouse, and I'll explain how that works later. And while that maybe didn't work quite as we had hoped, what did emerge is a new idea for a biomarker, which now we are trying to use in collaboration with a lot of the patients here in this group to see if that can be used for screening for OGT-CDG disease variants. Just as a reminder, sort of the state of the art in our lab as we know it now is we have actually now more than 70 OGT-CDG variants and over 80 patients. And by collecting a lot of data from these patients, and that's led by Marie, who is also, I think, listening in today, we are starting to get a more accurate picture of what this disease can involve. And I think we kind of started with intellectual disability, so all the patients that have that. But there's many other symptoms that we're now seeing that we didn't observe initially. So for instance, about 40% of patients have heart abnormalities, and about another 40% of patients have epilepsy, and that wasn't appreciated from the smaller sample size that we had. So by collecting more patient data, more variants, we can learn more about the symptoms, but also how maybe different variants give rise to different symptoms. So this is really important work, and it's great that many of you are participating. So just ignore the circles at the top. Basically, what the lab is really interested in is to try to understand whether this disease is a disease mainly of or during embryo development, giving rise to essentially an altered structure of the brain, or whether it is that the brain is properly formed and neurons, when they connect to each other in the brain, are not communicating properly. And this is an important distinction to make, because you can imagine that if this was a disease purely of things that have gone wrong during embryo development, it would be very difficult to correct that. Whereas if it's mostly a disease of errors in how neurons talk to each other, it is actually quite possible, and we have plenty of ideas on how this could be corrected. So we need to figure out which of these two it is, and that question will come back later. So the mouse model we've made is actually of the Truss family. So this is Eric, son of Ivar and Eve Truss, and I believe I saw Ivar listening in today. So we've now managed to make a mouse that has exactly the same variant of OGT that Eric has. And so Eric has a variant that sits right in a really critical part of OGT called the active site, which is where sort of all the magic of O-GlcNAc happens. And Eric has intellectual disability, general developmental delay, autistic features, seizures, dysmorphic features, and dyspraxia. And so we can now, using this technique called CRISPR-Cas9, we can make a mouse that has exactly the same sort of little mistake on the DNA that is found in Eric. So you could think of this as the Eric mouse, basically. And what we see in this mouse is that basically, if you look at panel G, this is where we look at the general levels of this O-GlcNAc modification. And on the left-hand side are 3 normal mice, and on the right-hand side are 3 different examples of this line derived from the mutation we found in Eric. So we can see that there is a loss of this O-GlcNAc modification, and as maybe has been discussed before, we can also see that at the same time, actually the enzyme that removes the modification called OGA is also almost completely lost in these mice. And so on the right-hand side is just a quantification of that. And I already want to point out here that in particular, if we look not at the protein level, but at the sort of bit of nucleic acid that sends instructions to make this protein called mRNA for OGA and OGT, if we take the ratio between these two, we have a very, very large difference. And this will come back later in the talk. So when we then look at what happens during development in this mouse, we see that about 7 weeks after birth, this mouse develops a weight deficit. So basically, this mouse is just smaller than the wild type mice. And this is particularly apparent in the skull. So if you look at panel B, on the left-hand side, you have a normal mouse, and on the right-hand side, you have a mouse with this particular OGT-CDG variant. So you can see that the skull is smaller. You can also see that the space that would be taken up by the brain in the skull is also smaller. So that is that endocast volume that you see over there. And this is interesting because smaller brain or smaller head is something that is quite commonly found in OGT-CDG patients. And some of you have seen this before, but with this very same mouse, now we see slightly stronger sort of movement defects. So this mouse is being asked to essentially climb on this bar and then go to one of the vertical where it could in principle slide down to the ground. And you can see that the mouse that has this very, very small error on DNA in OGT, really it's a tiny, tiny mistake. It is not able to hold onto this bar properly and move to the end. And this is even more apparent in a similar experiment where we take these mice and we make them face away from the table, and they have to turn around on this bar in order to walk back to safety of the table. So the normal mouse is on the left, and the OGT-CDG mutant mouse is on the right. So you can see that the normal mouse turns around very, very quickly, and the mouse with the OGT-CDG variant is kind of sitting at the end of the bar, and it can hardly make this turn, and then if it does, it actually falls off. So we can, this is of course just sort of graphically illustrated, but we can quantify this and show that really is a difference across a large number of mice. So that's kind of a new mouse. So that's already interesting for the field because it's only the second mouse with an OGT-CDG variant that does have symptoms that are a bit reminiscent of what we find in the patients. And at some of the meetings and in some of these fora, different types of treatments have been described or proposed. One is using inhibitors of the OGA enzyme. The others is a common nutraceutical supplement called glucosamine that many people take for rheumatoid arthritis, and that actually I know many of you have already gone and try it. It's a completely safe thing to try, but there is no scientific evidence whatsoever that this will help in OGT-CDG at this point. And then there's also this idea of gene therapy, which is that if we could introduce a normal, healthy copy of OGT using a virus, could we then rescue any of these symptoms that we see in the mouse and maybe later in patients? But the thing that we really wanted to ask is there a developmental component to this disease? So all of these treatments that you can see here, OGA inhibitors, glucosamine, AAVs, that is something that you would give to a mouse when, and indeed a child, when it's already born. But if the problem arises during embryo development, then you would kind of want there to be a rescue mechanism already during that stage of development, so well before birth. And so this is where we did an experiment where we made use of a mouse that we had developed about already 5 years ago, where we kind of simulate what would happen with an O-GlcNAcase inhibitor. So basically we have a mouse that has a point mutation in the really essential bit of the enzyme called the active site that makes it completely inactive. And in this case, the O-GlcNAcase gene, mice, and of course humans have 2 copies. And in this mouse, we have basically 1 copy that is normal and 1 copy that is completely inactive. If you make a mouse that has 2 copies that is completely inactive, then this mouse dies very, very shortly after birth. So clearly not having any OGA or O-GlcNAcase activity is not, that is not a good idea. But in this mouse, we can essentially have right from birth 50% of the O-GlcNAcase activity lost, and that is pretty much what you could achieve with a reasonable OGA inhibitor. So this is kind of like a way of simulating what an OGA inhibitor would do. So what we do in this case is we use the mouse that have this OGT-CDG variant that Eric has, and we use these mice that have one of the 2 copies of the O-GlcNAcase inactivated by a point mutant in the active site. And the idea would be that if we cross these mice, then we now have an OGT-CDG mouse where from the moment of conception, 50% of the O-GlcNAcase activity is lost. And so what we wanted to do with this is to kind of validate the use of OGA inhibitors to rescue some of the behavioral phenotypes that we see in these mice. I think the next slide is one with lots and lots of graphs. This is just to show that if we look at the levels of protein O-GlcNAcylation, then inhibition of 50% of the O-GlcNAcase by itself is not sufficient to rescue the levels of O-GlcNAc in this OGT-CDG mouse. So if you look at if you compare the gray column and the green column, then that is what would happen if you introduce the OGT-CDG variant in a mouse. And so that is kind of what you would like to do, is to rescue the green column back to the gray column. And that's what we try to do in the purple column. But as you can see, the purple and the green are more or less the same. So this looked like maybe that hadn't quite worked. But if we look at the ratio between the mRNA, so this is kind of the derivative from DNA that makes a message to make this OGT and OGA protein, we can see that that is partially rescued. Not completely. Again, you should compare the gray and the green bar to see what OGT-CDG does. And then the purple bar is what this partial rescue with this inactive OGA mutant does. And of course, the purple bar is absolutely not similar to the gray one, but it is definitely statistically lower than the green one. So this means that with this method, at least, we are introducing a partial rescue of this imbalance between OGT and OGA. That was nice. However, if we now repeat all of the assays that we have done, so this is basically just a spreadsheet showing all of the assays that we have subjected our OGT-CDG mice to. The first two columns are for the two different OGT-CDG mouse lines. The first one was published by us about 2 years ago. And the next one is the ERIC mouse that I've described here. You can see that when you make this cross that I've just said, we don't really get an improvement of any of the phenotypes. So what this paper basically shows is that, well, first of all, it's a completely new model of OGT-CDG with actually slightly different phenotypes than what we had in the first model that we described. It would be fair to say that 50% inhibition of OGA doesn't appear to be enough for rescue of any of the phenotypes we see in the mouse. But what this study also shows is a new idea for a biomarker. What I didn't describe is if you compare these two plots, the plot on the left is this ratio measured in the brain, but in the blood on the right is this same measure measured in blood. So that means that basically the levels of OGA and OGT in blood kind of mirror those what you see in the brain. And so this would then give rise to an interesting idea for a blood-based biomarker, which is what we are pursuing at the moment. And many of you have been in touch with Marie to recruit patients for this type of study to see whether this is indeed a marker for OGT-CDG across many different variants. And I think that is it.